For our first seminar this semester, we are happy to have Dr. Ewa Sell-Kubiak presenting for us. Dr. Sell-Kubiak is an assistant professor at Poznan University of Life Sciences, specializing in animal breeding and statistical genomics, with a primary focus on pigs and additional work in other species. Her research explores the genetic basis of reproductive traits in pigs, simulations of breeding objectives, GWAS, and genomic breeding value estimation. She actively participates in 2 EU Horizon projects, Meat Quality, where she serves as WP1 leader, and Breed Livestock, where she contributes as a WP3 scientist. Additionally, she is a management committee member for 2 cost actions, EU LIEHE and Insect IMP. Ewa also holds prominent leadership roles, serving as Vice President of the Animal Genetics Commission at EAAP and President of the Permanent Committee of the World Congress of Genetics applied to livestock production. And today she will be presenting on the genetics of trait variability and the importance of finding an optimum in production and reproduction in livestock. So I will turn it over to you, Ewa, and you can start whenever you're ready.
Thanks a lot for this introduction. There's not much more for me to say except for welcome to all of you, and well, I hope you will enjoy this talk, as it is a huge part of my scientific career, the topics that I will cover here today. So I hope you will find it as interesting as it was for me in the past years, and maybe sometime in the future I can tell you more about the other projects I'm involved, but for now I wanted to focus on this one. Okay, please, my presentation move. Okay, yeah. So when we talk about trace variability, you may come across different definitions, different words used to express it. The most popular are residual within individual phenotypic variation, genetic heterogeneity of environmental variance, environmental sensitivity, plasticity, canalization, and G/E interaction. Some of you maybe heard about some of those terms. For sure, G/E is the most common for you to know, and maybe it will be in this presentation given in a bit different context. All of those terms lead us to one thing, to resilience. And in animal breeding, the resilience is extremely important trait. And here, as by the 2 authors, how they establish it to define it properly, well, properly for all of us to understand it. So the capacity of the animal to be minimally affected by disturbance or to rapidly return to the state pertained before exposure to a disturbance. Okay, so basically we want the animal to be stable, right? We don't want the animal to be affected by whatever comes its way. And of course, the main reasons for that is economical, but economics, but not only this aspect. So, well, you are all working with some kind of genetics aspects of animal breeding. So telling about sources of variability in traits is for sure, well, very briefly needed for you. So basically, we can focus on aspects related to whatever happens in the environment, like globally, but we can think of environment in terms of smaller, so a farm at which the animals is kept, so different types of management practices, different types of feeding, and so on, and going even to the smaller type of environment, so the prenatal and pre-weaning environment that the mother gives to its offspring. But there is also a part of variability that is unknown to us, that is, well, in many terms, we cannot estimate it with any classical genetic models involving even more sophisticated analysis as epistasis or dominance and so on. Or we don't know yet that we should account for it in our models. And this is all of those factors are in general referred to as non-genetic. However, part of it goes to phenotypic variability in terms that I will show you today. And this part is actually more interesting than you can think about it. But when we think about these factors that we don't know how to exactly express with known factors in our models, we talk actually about most often about the residual variance in our genetic models. Every model will have the residual variance. We cannot avoid it. It doesn't matter how many fixed and random effects we will place in our models. But actually, this part is the part that is the most interesting for us when it comes to phenotypic variability. And why is that? Because it's actually it has actually a genetic component. And the fact that this residual variance exists and we can see it in the models, not only is there because we cannot account it with other known factors, but actually because part of the genetic variance is located in this residual variance. So if you want some proof, here is a long list of different traits with different species that this phenotypic variability was already studied. There were huge ongoing projects for many years with selection lines in rabbits and also in I lost mice. Oh, and in mice, as you can see, it's really long-term ongoing projects with selection to limit this variability. But you can see that it's in pigs, in fish, in different, in dairy and beef cattle, as well as in chicken. If you are interested, well, I assume most of you are really interested in dairy cattle. I would direct you to the PhD thesis of Marike Poppe from Wageningen University, as her thesis are very nicely describing how different aspects, different traits in that we can phenotypically record in dairy cows can be reflected later in this phenotypic variability, and with this in a resilience of the animals to the unexpected or sometimes expected environmental changes on the farm. Yeah, exactly. And the fluctuations in those traits can be used as indicators for resilience. And yes, definitely take a look into the thesis of Marike Poppe if you are interested in these aspects in dairy cat. Well, but what do we mean by this variability in terms of our animals? Well, basically, when we select our animals, we of course have certain best genotype in mind. So we want, in my case, all our pigs to be the same size when they are all grown and ready for us to, well, basically eat them, right? But we want them to be growing the same speed, to the same size, and so on. But of course, those environmental factors will be affecting the growth of the animals. And some of them, like some kind of viruses that can happen and don't affect too harshly the herd, but can affect the animal itself, will reduce its growth, for example. And having a perfect genotype describing low phenotypic variability would show us those animals that can be resilient to the fact that these environmental factors affect them and that we can maintain them on the level as expected based on their perfect phenotype for that we have in mind. Well, but we know that there is of course a genetic variation, as I showed you, in multiple traits, and we can track it to different species. But the question is whether we can see also the genes that are controlling the phenotypic variability. In our classical approach, the QTLs that control genes are linked with the mean of the trait. So the fact how many, what is the milk yield, or how many piglets we have, and so on. So we are focused on the average or level of a certain phenotype. When we're talking about genes that control the variability, they are often referred to in the literature as VQTLs to distinguish them, as they are focused on variation, so how many differences there is between the animals, rather than the mean of the population that we would like to, that we already know about. And how should we understand those VQTLs? Where this is a proof that there is a presence of unmodeled interaction that is associated with the locus. And this interaction can be G by E, which is for you, for me as well, the most common type of interaction. So it can be caused by the environmental factor that is known or unknown. G by G interaction, and parallel presence of both of those interactions can also be affecting the presence of the VQTL. The most well-studied genes that were very highly published in Nature and are linked with variability are one SNP that is located in FTO gene linked with the body mass index in human, and also a gene called HSP90 that in Drosophila and Arabidopsis was shown to buffer environmental factors that can affect the morphological appearance of the fly and the flower, and the most important developmental traits. So how should you understand it? So basically, this gene, if there is something happening in the environment that would cause the fly to have the uneven wings that would stop the fly from flying, this gene will block this kind of effect of the factor, making sure that while developing, the animal will have a proper size of the wings for it to fly. And well, similar with the plant with the same number of petals or shape of them. And there's again lots of different species in which the VQTLs were already established. So you can see here even plants. So maize even is on the list. Rat, the laying hens, dairy cows again, of course, beef cattle, and rabbit. And when preparing this overview of different traits and different species that you can already take a look into different VQTLs, I've noticed that there's more and more studies involving VQTLs in human. So this just as fun fact for you. So I knew about the study on inflammatory biomarkers, but there is also follow-up study on body mass, but also blood traits and absorption of vitamin D in human. That is all having detected clear VQTLs for the variation between humans in those different traits. So clearly, it's very interesting not only for the animal breeders, so in livestock, but also in human studies. It becomes an interesting aspect. Why do we now? Maybe it was a brief info about humans, but now, of course, going back to animals, we will not control the variability of traits in humans. I hope it will never happen. But why are we interested in this when it comes to animals? Well, we're thinking about animal welfare, health, and of course, as I mentioned at the beginning, economic efficiency of animal production. How the welfare could be affected by those differences? Well, extreme cases of any trait can affect the well-being of the animal. Too large litter, too large egg, and so on. And the animal itself is only one individual, and in terms of the whole population. So on average, we might miss it, but when you look at the variation, you can see those outliers, the very low values, which are not good for the farmer in terms of economics. But then you have the extreme values that will affect the animal. And actually, in Europe these days, we have a lot of discussions regarding setting an optimum for different traits in animal production. there was a big discussion, rather heated discussion at EAP last year regarding that, as the lobbyists are really pushing that there will be certain limits that the animals shouldn't produce more than. This is, of course, very utopic way of thinking about animal breeding, but since it becomes so important issue that there is whole 4-hour panel at such a large conference as EAP is, which gathers 14,000, well, no, 1,400 people, not 1,000, absolutely not 1,000, 1,400 people. That shows how important it is to think about it, that there are certain limitations that the citizens will push on animal production and livestock production in general. Well, but I'm expert in litter size. So from now you will hear only about litter size and, well, the importance of looking into the limited variation in this trait. Well, so economically speaking, it is the most important trait in pig breeding. And if you talk with breeding companies, doesn't matter if it's PIC or Topics Northfin or Dunbred, whatever, they will tell you that the more piglets, the better. Of course, there are certain aspects involved in breeding of larger litters, which is making sure that the number of stillbirths is decreasing, because that is a big issue. However, in general, the breeding companies still think that we can go further, we can push the animal further. And well, there's plenty of different SNPs and QTLs detected for litter size. You can see them in PQTL database. I think there's several, it goes, I think, almost to 1,000 right now. There's really huge interest in finding genetic background for this trait because of economic importance of it. But what we observe is high phenotypic variability between sows and also within a sow. So how many piglets the sow has in different parities. So in my opinion, but as I showed you, not only in my opinion, there is certain need for an optimum of litter size. What is this optimum number? I don't know yet, and I can guess, but we should definitely think about it and to maintain this level. And why? It's because of sow and piglet welfare. So for sow, if the sow has very large litters, it affects its health. So the joints can be damaged because of the heaviness of the pig during the gestation. Also, the lactation is very demanding if the sow has nearly 20 piglets. And for piglets, the bigger the litter, there are issues with delivering such litters, so there are problems with having all the piglets alive at the end. And also the space that the uterus has in pig, it's not unlimited, right? So not every piglet can grow with to the full size because of that. Well, and of course it affects farm management and economics. So no farmer wants to have piglets that they cannot sell later on. So it is important that majority of the piglets are born alive and healthy. And if the sow has very large litters, sometimes it's necessary to switch the piglets between sows and provide some artificial techniques to feed those big litters, because simply the sow doesn't have enough nipples or is not capable, not physically capable to maintain lactation level to feed a lot of piglets. So when I say about variability, what are in terms of the averages for litter size? Well, in large white, and 95% of the information that you will see here is about large white pigs. Those litters can go from 1 to 28 piglets, but I already seen in some newest databases that there is the number of piglets goes up to 35, 36. And I'm talking here about total number born. And in pig breeding, total number born refers to the all the piglets that were born, but not necessarily were born alive, because that's a different trait. So it accounts for stillbirths. And on average, in this pure breed, we have 13 piglets, and with standard deviation of 3.5 piglets. So there is huge variation just looking at the standard deviation among the pigs and well, among the litters actually. So based on my work still from my PhD times, I knew that variability of litter size is heritable, and it can be changed by 9% after one generation of selection, assuming perfect conditions. Of course, this is for let's say simulation type of information, but it's very idealistic. What we know is that genetically speaking, so based on the genetic correlation, litter size is when it's increasing, then the variability is also increasing. So actually, for the years of selection to have more piglets in a litter, we also increase the variability. So the fact that we see such small litters and very huge, very big litters is actually linked with the fact that it was ignored to look also into the variation of a litter size, not only into having more piglets. And phenotypically, in terms of the phenotypic correlation, we actually see that bigger litter sizes will lead to smaller variability. And in this work that I'm referring here to, I also showed the very first SNPs associated with variability of litter size in pigs. As you can see, not many SNPs in this GWAS study pop up as significant. Actually, only this one triangle, red triangle, was the one that was fully could be fully accounted as a significant SNP. It's, as you can see, it's not p-values as you normally see with the GWAS studies, but Bayesian factor, because I used here a program that used Bayesian statistics to get this GWAS. And I got there quite interesting candidate genes on this chromosome 7 for variability of litter size. But the one that was for me the most interesting was the fact that the Suscrofa version of the gene that was already published and proven in Drosophila and Arabidopsis, I actually found that this gene could also be involved in the variability of litter size. As you can see, because there are 3 genes shown and only one SNP was available, that means that I looked into larger spread of SNPs, well, of the region before and after this SNP, because exactly in the SNP was not located in any known gene. But anyways, that was very interesting result, and I wanted to see where can I go with it further, because of course candidate genes don't mean that there is causative mutation. And I decided to write my own project focusing on genetic mechanisms regulating phenotypic variability, and hopefully finding the causative mutations behind the pigs that are very well prepared for being well, resilient to different environmental factors. And for this, I received a grant. It was fully funded by the Polish National Center for Science, but I had data from Topics Noordsveen from Netherlands, and because of my PhD from Wageningen University, I was still in contact with my promoters. So it was a cooperation between 3 partners. And as you can see, it took a really long time to do all the studies, but different reasons were behind it. But anyways, the important is the outcome. So of course I had some objectives for this project. First of all, I wanted to really take a proper look at different models used for to establish phenotypic variability, and then of course move on to looking into new associations or maybe verifying what I already knew from my PhD study, and then moving to identifying genetic variants that would be responsible for causative mutations. Well, so now a bit about the models. So this beauty here is a bivariate model. I hope you all know how those things look like, but it's a special bivariate model because it's double hierarchical generalized linear mixed model, which actually does a trick like this, that it takes the residual variance, well, actually residual per observation that we get from the level part of the model, and places it in the as a y variable for the second part of the model linked with the variation. So this here it looks way simpler than all the coding that went into developing this analysis. But what was here an issue was the computation time, because it lasts for several days depending on the scale of the data. So the more data, the longer it took to analyze it. So I was thinking about using simpler method. So here you see well, very simplified animal model for litter size, and of course we have here our residuals. So what I did here is actually that from whatever program you use to estimate variance components, you will get residuals per observation. So here you see example, right, of 2 sows. For each of the record of a sow, I got this residual, and then that was the result of a classical analysis. And then I took a variance of those residuals per sow, transform it with logarithmic transformation to obtain a normal distribution. And this allowed me to get the variability phenotype. This is very simple. The regular animal model takes what, like 5 minutes maybe, an ASREMO, and then having this adjustment done and obtain new phenotype that was later analyzed again in a separate model was again some, well, let's say 20 minutes for preparation of the data and 5 minutes for running. So let's say 1 hour instead of several days or even a week or so. So for me, it was very important to know that this new procedure is faster. The new phenotype of variability is heritable. I didn't show you this here, but that was the case. And there was also the well, I again showed that there is positive genetic correlation between litter size and its variability. So I wanted to make sure that it is possible to select the traits in a desired direction. And here you see the ellipse of possible economic values. Well, not economic values, the response in litter size variability on x-axis, and on y-axis you see response in average litter size. And actually only the economic values that I gave in the simulation to the estimate the response, only this region here provided me with the values I wanted. So reduce response in litter size variability, at the same time, no change, or increase in average litter size, because as I mentioned, the breeding companies still want few more piglets. But then I also thought, okay, but we are keep on focusing on the entire population, but maybe we could also see what's happening within a sow, and maybe it can be the variation can be accounted on the level of a sow. And the idea came from the fact that when looking at the average litter size per parities, so here you see on y-axis parities from 1 to 10, and well, on y-axis is the average litter size. I saw that actually resembles the lactation curve. So I tried and fitted the Legendre polynomials, and it appeared that it actually, you can really nicely account for this curve representing different litter sizes across the parities. And the question was whether we can flatten it. First of all, I checked whether there is genetic variation, and this whole gray area around the curve is actually this genetic standard deviation. So you can see that there is huge genetic variation around this curve. So clearly you can play around with it and try different things. But what we observed was that if we, for example, decide to have more piglets in a first parity, the entire parity curve is increasing on average, but it doesn't change the shape. And when we decided to put no pressure at all on the first parity and make sure that those animals are not pressured too much into having even more piglets, then the whole curve decrease. So basically we lose piglets all over the place. So that it's absolutely not something that we would like to do. So basically, okay, I showed that it is possible to play around a bit with the parity curve, but we cannot really go further with changing it. So yeah, so this information showed me that there are different ways of account for phenotypic variability in litter size. There are different methods. The models can be extended and extended and extended, but in the end, we need to look for things that can lead actually to implementation in animal breeding, and definitely this bonus study was not giving that. And importantly, we saw that it is possible to select for increased litter size with the reduced variability if we account for this other trait in a breeding program, because that's important thing. If we keep on continuing increasing litter size, the variability has to be accounted for. And on top of this additional thing, we saw, well, I saw also one more aspect. I don't know if you ever heard about something called second litter syndrome. So basically that's situation when the animals in the second parity, they have less piglets than in the first one. And well, here on this curve, you see that those were really highly producing animals in the first parity. So maybe they were overstressed going into second one. But actually, thanks to this study and follow-up study on the second litter syndrome, I showed that actually whatever we see is actually linked with the normal distribution of litter size and the genetic variation around the parity curve. And it shouldn't be called a syndrome, to be honest, but from 2001, nine more papers appeared about this second litter syndrome without completely ignoring the fact that I showed it doesn't exist. But what can you do? It's good that they are citing me. And then after having all those different, let's say, classical quantitative genetic models done, I wanted to move on to looking into genetic background in terms of GWAS study. So here in this table, you see a summary of information that I had in my study in 2015. So when I was finishing my PhD, and here how much data I got extra in 2022. And what I did in this study, I decided not only to focus on this because this complicated method, well, maybe not complicated by taking a lot of time in computation-wise, but also I decided to use the, let's say, simple way of obtaining variability phenotype, and also this more complicated way. And based on the fitting the models, I already then saw that those two methods are not the same, and actually the simpler method is causing such a ranking of the animals in terms of EBVs that the accuracy is reduced, and also it's more difficult to predict what to expect from the next generation if you use the simpler method. Okay, but with this knowledge, I went into GWAS study. Again, huge increase in numbers. The fact that I had here such a large number of SNPs and animals genotyped was of course partially possible because of the imputations of the genome. So well, that was very helpful for sure for the breeding company, because it cost us less money. But for me, it was also good to have more data, because the heritability of the trait I'm interested in is very low in a classical sense. And in both GWASs, I also used sires, not only the sows. The fact about it here was linked with that there was very few animals genotyped. Well, very few. Now we know it's very few, but back then it was quite a huge study. So here the boars were also included. So I wanted to repeat it in this study to make sure that it's really overlapping with results. So what we saw for the, let's say, phenotype from the simpler method, there were 3 regions highlighting as with significant SNPs. For me, the most important was that, again, I see something on chromosome 7. But when I moved on to the analysis for the more complex phenotype, well, more complex phenotype coming from a more complex method, only this one SNP remained significant. So chromosome 1 and this other, what was it, 18 was not, the SNPs on those chromosomes were not significant anymore. So again, it was a proof that the methods are not the same when looking simply into predictability and EBV value estimation, but also when we looked into the GWAS results. And of course, my question was whether the 2 SNPs are the same, because only one SNP significant, different methods used, but way more genome coverage available and so on. But unfortunately, those were not exactly the same SNPs. And we, with my co-authors, we decided to check whether the issue was with the reference genome. But because there was a new version in the meantime, but the location of the SNP from 2015 changed a bit in the new reference genome, but even went further away from the new SNP. But LD between them was quite significant as 0.11. Well, but still, what was very important for me is that the chromosome 7 was again linked in this study as the most important for phenotypic variability of total number born. And this time the SNP was located in genomic evolutionary rate profiling region, which is apparently extremely conservative region. And if there are any mutation in regions like this, it can cause some real problems for live creatures. Well, I don't know much more about it, but this is what the experts told me. And there were some potential other genes as candidate genes close by, let's say. So overall results of all this work were that there are multiple ways to study phenotypic variability in litter size of pigs, that the methods, even though they kind of look similar in terms of what they're doing, they are not really interchangeable, and it is important to know what are the limitations of the simpler method. In large white pigs, chromosome 7 is the most important for litter size variability, but none of these studies led me actually to show indicating causative mutations, because I couldn't really see proof in the phenotypes when it came to looking into differences between the animals in the genotypes that were the most associated with the trait of interest. As an additional thing, I want to show you that in other breeds, the regions are completely different. This is a study done by my MSc student. Now she's working with me still on her PhD. But Gabriela did the same, but on in landrace pigs. And as you see, in her study, chromosome, 3, 4, and 13 were the most significant. Oh, I skipped here chromosome 6 as well. Okay, but her study showed that the data in landrace are completely different than in large white. We actually needed to do Box-Cox transformation of the data. This is this small BC you see here, and this had to be done to adjust the phenotypic data to actually have a normal distribution and so on. So there are many different aspects of the variability between the breeds, even within one species. Well, you never work in such projects by yourself. So here's a list of all the people from different groups that helped me out. So I'm still working with most of them. So it's very good that this cooperation continues, even though time flies and there is big distance between people. Okay, that's all for the study part, but I have some advertisements for you. None of them is about World Congress, but maybe I will interest you in some other things. As Ella mentioned in the introduction, I'm involved in cost actions. Cost actions are the initiative of EU Commission, helping people to network, transfer knowledge, educate, and brainstorm. Basically, this is the purpose of those cost actions. I'm involved in two. This one is probably more linked with what you're working on. It's European Network on Livestock Phenomics. We're actually focusing on gathering all types of information linked with any kind of phenomic data you can collect on animals, and trying to see how we can combine it all and use it all in livestock. This is European initiative, but you can still become part of working groups, and with this you can get information about workshops, about some meetings, lectures online, this kind of things from different people involved in this cost action. And also from this year they changed the rules when it comes to scholarships. So you don't need to be from certain EU countries to actually be eligible to apply for scholarships to go for some training schools or conferences or even short-term visits. So if only you become a member of a working group, which actually doesn't force you to do anything, you basically just sign up for newsletter, you might try to get some funds for something for you if you need that, of course. And second cost action I'm involved in, it's very nice for me because it's completely different. It's focused on insect breeding. And I was asked to join it because of my knowledge on animal breeding, and they needed somebody with a strong genetic background in livestock, and also, well, my network links tied, good links with World Congress and EAP in Europe. But if you're interested in knowing anything about insects, you can again sign up for working group and simply just, you know, see what's going on there. EAP is now organizing a workshop, first ever workshop on insects in Greece this year. I was asked to give a crash course on quantitative genetics there. So in 2 weeks, I will be busy doing that for international crowd. But for me, it's also a great opportunity to learn also some stuff about insects, because it's completely new, and sometimes my ideas how they can improve the breeding objective is not working, because the insects are very small and it's very difficult to collect individual data on them. So that's just as a fun fact for you to maybe join this. And I also wanted to really encourage you to force your supervisor to send you for the conferences, because my experience is that conferences are really the best way to network, to get people together working on similar topics. And thanks to the conferences and my Twitter account, which doesn't have too many followers, but still, I met this nice crowd of people from Spain and Switzerland, and it helped me get back on working with birth weight uniformity in pigs. And well, this is ongoing for quite some time. We are now ready to publish a paper out of it, but just to show you that it is important to maybe get scholarship if your supervisor doesn't have the money, but also try and visit as many places as possible. And for sure, World Congress in 2026 will be great place to show your studies, show your research and network, and maybe get options for postdocs and other work in any part of the globe. Well, thank you very much for your attention. Oh, 2 minutes over time, so not too bad.
No worries. Thank you very much, Eva. That was a very interesting presentation. We're going to be opening up the floor for questions here. I see some hands, but I'm going to start with Dr. Christine Bays put a question in the chat, so I'll start with that. Do you also look at within litter variance of piglet weight, and how is that associated with litter size variance within and or across sows?
Yeah, I did one study on, well, actually 2 studies, but back during my PhD. Now I went back to that with the cooperation with the Swiss and Spanish people. Yes, there is a huge link, and breeding companies are not really willing to show the results of it, because it's very economically important trait, the birth rate variation. So the correlations are not really available. But what we observe is that if we focus on litter size and increase uniformity of litter size, we also see a change in birth rate, and other way around. So it is beneficial for those 2 traits in a good way. It's beneficial in a good way for those 2 traits.
Next, Filippo, you want to ask a question?
Yeah, it's actually more a comment. I want to thank Eva, because also reminding us about this cost action, which in the past we've been, I remember Metagene project, and it was kind of help us in our strategy about the methane. Was great. I think a couple of our student researchers went to, or they do also training, if I remember correctly, within this cost action, and went to that, and I went to the final thing. So thank you for reminding me about that. I will definitely follow up with you and Luca on this ULIFE. So I appreciate that.
Yeah, it's also a great chance to actually network in terms of having the projects, joint projects, build consortium and so on. In Horizon projects, we can involve people coming from outside Europe as well, if it's beneficial for the project. So it makes a lot of sense to get to know people like through cost actions.
Thank you.
Yeah. Flavia, would you like to ask your question?
Okay. Can you hear me, Eva?
Yes,
Yeah.
very well. Hello.
Yeah, thanks for the presentation. Very interesting. I have two questions for you. So you showed that you focus on purebreds, right? So then you look at this variability of the litter size. Have you looked at how this would compare with the variability that you see in the crossbred animals, the commercial ones, that are the results of the crossing of large white with Duroc, for instance? Because if I understand, my expectation is that you're going to have less variability in the F1, that you will be the commercial product because of heterosis, right? Have you checked on this? How the variability that you have in the lines that are crossed would reflect on the commercial piglet or commercial soy in the litter?
So the sows are purebred, indeed, large white, but sometimes the litter was not, because it sometimes the litter came as a cross, but it was accounted for in models as a fixed effect for the fact whether the litter is purebred or cross. But I've never worked with actually the crossbred sows producing litter. I never got access to that. My study on birth within litter variation was on crossbred sows, and well, no, that was also a litter. No, so I never got access to actually crossbred animals that produce litter. So sorry, but I don't know what to answer.
Yeah,
But you're probably right that the variation would be lower, right? Because it would.
Yeah, basically, then is related with my second question. So you are look at this residual variation, right? That genetic variation that's in the residual of the model. But how that variation would compare with the Mendelian sampling variation? Because why I'm asking this, because most of it's very difficult to have a uniform progeny because of the Mendelian sampling, the Mendelian variation. And it's very difficult. It was impossible in the past to select for uniformity because we couldn't control genetic variation. But now with the genome selection, genetic markers, we can select for less recombination, for instance, less variability, right, on the Mendelian sampling. So again, Eva, do you have idea how much you can gain in that direction, like having better control or now exploiting for less Mendelian sum variation in your approach of look at what we don't explain with the models?
That's a very interesting question, and I don't know the answer to it.
Okay, so that's the idea for the next project.
Yes, clearly. But I will ask my PhD student to do that.
But
That will
at
be
least to compare the amount that you expect of that you can change of the variation, right?
Yeah.
By selecting for smaller variation of the Mendelian sampling or controlling the Mendelian sampling in exploiting the variation that's in the residuals, right? Too small.
Yeah. So we, of course, the variation that you get on variability, on variation, genetic variation, it's logarithmic, and so we did translate it, let's say, to the level, and that allowed us to show that in terms of ideal breeding program, you would have a change of around 9%. So I think it in a way reflects your question. I think that because that would be the maximum we could achieve, and that would be the closest to link with the Mendelian sampling expectations of the variation, I think. Yeah, but definitely something to look into more closely in quantitative genetics way approach. Let's say. Yeah.
Yeah. Thanks so much, Ewa. Great presentation.
Thank you.
Thank you, Flavio. We have a question here in the room.
Yeah. First of all, Ewa, congratulations. My question is because you found different chromosomes in your GWAS output for landrace and large white. So as you found different chromosomes, different SNPs, and probably different genes surrounding these SNPs, but have you checked the functional aspect? Because sometimes different genes are involved in the same pathway, some biological process. So have you checked this?
We didn't do that with the previous studies, but right now, Gabriela, my PhD student, she's working with the GWAS using the sequence data, the full sequence data, and we're hoping that this will give us more information, and we will be actually able to go deeper with the molecular basis of this trait. But we already see she went back to work with the large white sows, and we already see that there are different regions popping up. It's not chromosome 7 anymore, which is a bit problematic, but that might be caused by the fact that now we don't use the bores are not included anymore in our analysis, and maybe this chromosome 7 was popping up because of the bores involved, included in the GWAS in previous studies. So there is clearly a lot to be done, and this aspect that you just rose, it's Martijn Derks from Wageningen University and Topics Norsving. He's helping out us with getting deeper with the answers, because my expertise is not that molecular, so I need somebody to help me out and to actually go deeper into those processes.
Thank you.
Do we have any more questions online or in the room for Dr. Sell-Kubiak? One question is everyone else does. I was wondering what degree of precision you would expect if you were to select for both something like litter size and the variability of litter size. Would you expect to be able to have a line of pigs that are able to consistently produce, let's say, like 18 piglets every litter, or would it vary a lot more still, even with that selection?
Well, I think we would be able to get the best of the both worlds, but it will not be an easy process. At the same time, in Switzerland, for example, they completely skipped selecting for litter size. They just maintain it in the breeding program. They give it the economic value of 1 and just maintain it as it is. So there are ways to actually think about it and indeed select still for increased litter size and lower variability. And I think it is something that can be achieved, as the breeding companies achieved with the decrease in the stillbirth number, because that's possible, even though the correlation is also there positive with the litter size, and they can still get bigger litter sizes with the decrease of stillbirth. But what we see is actually that some companies decided not to increase it anymore, and it's not affecting negatively the production of those the numbers that they get on the farms when it comes to litter size. So I think there are more ways to look into that, and but in my personal view, we should really focus on the optimum and try to achieve this this level, not pushing further the animal, let's say. I know that didn't exactly answer your question, but there's just different options how we can look into that.
That's very interesting. Thank you. Okay, I'll make a last call for any questions before we close it out for this afternoon. It looks like no one has any more questions. So sorry.
I'm curious now about something. Eva, for this large white population that you analyzed, how many pairs of teeth the sows they have?
The average is 16, but it's the functional teeth. They really pressuring the large white and topics northern to have more and more nipples, and it's causing the peak to be longer actually.
Yes, so that's because the heritability of the number of teeth is moderate. So it's possible. So that's another component, right? If to balance the litter size, you can select for teeth, but there is genetically correlated with number of vertebrae, right? So these longer pigs, they have one extra vertebra.
Yes.
So it's quite interesting because the animal is really changing to accommodate for being longer. And so, but then you work with 14, you said, like 14 or 16? 16?
16, yeah.
16.
It's really increasing fast, but the issue is with the number of functional teats. That's the problem, that the rear ones not necessarily produce milk. So it is important to count the functional teats. Yeah.
Okay, thank you.
Very much, Eva. I think we will close it out for this afternoon, but thank you so much for your presentation. It was very interesting. The discussion was interesting as well. And we will thank you. And we will see everyone here back next week for another CGIL seminar. Thank you, everybody.
I